Good afternoon, ladies and gentlemen. Today I'll be talking about something of the things that I have done in my past, a module that I have formed myself without much of my knowledge. Because I have done it, I did not know I wanted to do it, but it has happened somehow or other. So the heading or title of the talk has been kept as research module. Of course, this Ratna has, I think, framed the name. Research module in reproductive medicine and lifetime experience of a clinician from stethoscope to microscope. In fact, I wanted to be a clinician. I did not know what I did want. I wanted to be a good doctor. And that is what I wanted. But ultimately, I find that this is a lifetime experience of a clinician from stethoscope to microscope, and that is exactly correlated, which I did myself. At the end of the talk, you will appreciate. And in the morning, I was just doing a dummy talk, and my students were there. I saw this is exactly the phases I have passed through in the different periods of my life, and it has correlated with the performance that I have done, good or bad, small or big. Now, the title means that stethoscope depicts physicians involved only in clinical diagnosis of a disease and its management. Microscope indicates physicians also involved in understanding the process behind. That is, 3 points there: genesis of disease and its prevention, if ordinarily not prevented or curable, to find out ways by which it can be prevented or it can be cured. Of course, the journey from disease research to disease cure is not generally a very smooth one. It is very difficult, as you will see at the end of the lecture. A clinician-scientist combination or a researcher is the person who is the bridge between the laboratory and the clinic. He or she is a doctor who can correlate the disease process both in the patient as well as in the laboratory. Very difficult, but in practical terms, it is difficult to integrate these two practices in one setup or in one individual. What happens in the other countries? I say, still our developing country, we have already developed, but in other countries, in all teaching hospital, there is an experimental wing to bridge the gap between the clinical wing and the central and the research wing. The gap between the clinical and the research medicine is bridged by these experimental units. And in our country, there is a wide gap, and it is still persisting and it is still existing. Now, for example, in India, such facilities rarely exist. Busy clinicians rarely find time to refresh and update their knowledge through periodicals and journals. The only source of refreshing memories and addition to the knowledge, recent knowledge, is through pharmaceutical representative. They come and say, this is the medicine, sir, this is the action. Only they do not go to find out actually what is happening in the world, but they only know, only for 5 minutes the representative talk to them. But further research is essential to improve clinical medicine, because application of effective clinical medicine entirely depends on a sound-oriented theoretical background. And a clinician actually who is treating the patient rarely finds a time to go through those periodicals who are supplying the information, and therefore the gap remains there, persisting there. Let us talk about Robert Edwards and Patrick Steptoe, the pioneers of in vitro fertilization. Both of them started their medical career with stethoscope, but finally one, Bob Edwards, entirely switched over to microscope, and the other Steptoe to laparoscope. Now, Edwards totally diverted to reproductive physiology And step 2 initially limited himself to visualization of pelvic organs with laparoscope, and finally switched over to follicular physiology and tried to find out the exact time of oocyte retrieval, because the oocyte comes out when you don't know. Unless you can get a metaphase through oocyte, well, your success in fertilization of a fully grown oocyte remains unsuccessful. Therefore, the exact time of oocyte retrieval in IVF laboratory was found by Patrick Steptoe by keeping the laparoscope inside the abdomen for long hours to find out when the pinkish color changes, and that is the exact time after 34 hours to 42 hours after the LH surge, and that is the time when to pick up the egg, and he will get a mature egg, and that is how he got a success in getting a mature egg. There was no LH at that time, there was no RIA at that time. It is only by your mere perseverance, it is mere merit, it is only by your mere thought that you have to find out the ways by which to know these things. But prospective physician, every prospective physician at the time of entry to medical curriculum or thereafter have this rare instinct for deviating from stethoscope to microscope, or is it desirable or good for everyone and the society? Certainly not. It all depends on the genetic drive or the environmental module that drives an individual, either whether he will become a clinician or he will become a scientist. And that is the combination of genetic influence and environmental. Sadhu hoga to acha sange thakle sadhu hobe, kharap sange thakle dakat hobe. So that is the dictum. It has been operating years after years. For example, it was presumed that by cloning, যখন cloning science বেরোলো, that is in the year 1997 I think, yes, by cloning celebrities could be duplicated. C.V. Ramanকে যদি ক্লোন করা যায় তাহলে C.V. Raman তৈরি হবে, কিন্তু তাকে হবে? Actually it's not a fact. Subsequently this idea had to be abandoned, because by cloning one can duplicate phenotype, that is the physical appearance of the individual, but not the functional potentiality. The functional potentiality, what C.V. Raman had, what Rabindranath Tagore had, that is by cloning that cannot be duplicated, because it is by the specialization. Something else is essential for becoming Rabindranath Tagore, a Rabindranath Tagore. Not Ram, Sham and Yodhu, Madhu can be. Of course, this is apart from other factors were there, for which there was a strong debate whether by cloning, where a celebrity can be kept alive for years together, for months together, for centuries together. It was not a fact. It is only the physical appearance that you can clone, but it is not the functional integrity of the individual that cannot be cloned. So that was a fact. Why is it so? We all know that all cells of the organs and tissues of our body have three functions. Now we are coming to the science. Now there are three functions of a cell, and cell of the human body, and cell of the human tissue, or cell of the, typical classification of the three divisions. And these are division of the cell, differentiation of the cell, and specialization of the cell. Meaning thereby, we know in physiology a cell divides, nucleus divides, and after that the morphological appearance also divides. Cell divides means nucleus divides, nucleus divides means the cytoplasm is also dividing, and differentiation means a normal neutral cell which will come just now, stem cell, this is the reserve cell, and that will change its function. Function মানে একটা হচ্ছে the liver cell, একটা heart cell. They came from the same cell, and from the same cell gradually the shape changed, the appearance changed, and then the function changed. A cell, that same cell which we call will come now, which we call the stem cell or the totipotent cell, and which can form any cell of the body, heart cell, pancreatic cell, liver cell, pancreatic cell. So every cell in the body came from the same cell, a fertilized egg and a fertilized mother. It is very surprising. But when they become a heart cell, the same cell, the fertilized zygote, when they just go on dividing, they gradually change in appearance also. The hexagon, pentagon, all these appearance also gradually changing. And at the same time, they are changing from the neutral, undifferentiated cell to a specialized cell, that is a liver cell. That is a hepatocyte, a cardiomyocyte. When the undifferentiated cell changes into hepatocyte or cardiomyocyte, they do not know each other. They were brothers and sisters, but after they become adult cell means somatic cell, heart cell, that is, they do not understand or they do not recognize their sister, that is the cardiomyocyte or the hepatic. So how it is happening? Division, differentiation and specialization. Gradually they are changing not only changing their structure, not only changing their appearance, but also changing their behavior also. And division means daughter cells divide but maintain the morphological appearance of the mother cell. Differentiation means daughter cell deviate from the basic morphological appearance of the mother cell. Specialization, daughter cell deviate also from the functional behavior of the mother cell along with morphological appearance. So when the daughter cell of the liver asks who is the daughter cell of a cardiomyocyte, that is the heart, well, you ask him, no, I don't know him, but originally they had the same origin, which in one term, stem cell term, it is known as totipotent, and in the name of the anatomy, it is known as the adult stem cell. The adult cell, they become liver cell, they do not understand or they do not recognize the mother cell. So this is I can't show you the picture photograph, but you can see the photograph. The sperm and the ovum united together forms a zygote, and from zygote it comes to the embryonic cell. From embryonic cell they come to the morular stage. From morular stage they come to the blastocyst stage. In the blastocyst stage they differentiate, totally differentiate. One group of cell become remain as the embryonic cell, other group of cells, and they will form the entire body, entire cell mass of our system, and they will not recognize the other group of cells, which is known as trophectoderm. They will form the placenta. So placenta is different from trophectoderm. The differentiation of the functionality will change totally from there. So one group is the dividing, differentiating and functionality. All these things will be different from the placenta. And all the tissues and organs of our body will form from the inner cell mass, that is the blastocyst. So the one group remains, or it is also known as inner cell mass. Inner cell mass group of cells will form all the organ, tissues, fluid of the part of the body. And the other part is the placenta, which is not a pluripotent cell, a totipotent cell. So one group of cells is known as pluripotent. They are helpful for forming the cell, and they are the precursors of the embryonic plates. That is, there are three embryonic plates. One is the mesodermal plate, one is the ectodermal plate, one is the endodermal plate. One will form the brain, heart, lungs, etc. One will form the kidney, bladder. So all groups, when they have become adult somatic cell, when they go to the embryonic stage, each cell of the body has got a difference in the potentiality, functionality, morphology, and rate of division. So this is how this happens. Why this happens? And some of these cells, because the cells die, cells grow, cells divide, and they are replaced by new cells. And this new cell, some cells are kept reserved. So these new cells reserved cells are known as stem cells. So there are 4 types of cells at that time. At the end of this game or play, it is going on playing, and division, differentiation, and specialization. They are specialized. I am a gynecologist. He is a physician. Originally, both are doctors. But when you are working in the laboratory or when you are working on the patient, we are completely different. We do not recognize. So that is exactly what is happening here. So that is, you can see here, so this origination or origin of the different type of tissues of the body. And the tissues of the body have been renamed. And what is the renaming of the tissue? That is the ectodermal, mesodermal that we have already said. And some will remain their own morphological structure, groups of tissues or cells, groups of tissues and cells, and these groups are known in three terminologies. And there are some reserve cells. In each group there are some reserve cells. So one group of tissue is known as your embryonic stem cell. And from that point of view, there are three groups. One is the endoderm, and from here the brain and skin are formed. The one is known as the mesoderm, muscle, blood, etc. Now there are three broad groups: ectoderm, mesoderm, and endoderm. And the fourth group has come in, that is the germ cell. There is a big difference between this ectoderm, endoderm on one side and the germ cell on the other side. The germ cell cannot divide, and there is no mitosis. Divide means mitosis. The nucleus and the cytoplasm are different. But here in the germ cell, there is only meiosis. Meiosis means two daughter cells are not created. But in the germ cell line, only one daughter cell is created and the other is rejected. That is what is the meiosis. So meiosis is not creation of two cells. Meiosis is creation of one cell, an adult cell, and that is only limited to the egg in the female and the spermatozoa in the male. So this human embryonic stem cell is from inner cell mass. There are some special cells which are kept as reserve cells. When the cells are becoming deficient due to your wastage, cell wastage, which is or we can say it is apoptosis. When the cells undergo apoptosis, well, usually they are replaced by the stem cell. And the stem cells are eternally surviving, eternally being revived and being replaced by those which are becoming older. Now, after differentiation of somatic hepatocyte, the peculiarity is, so far as the stem cell, adult stem cell or adult mitotic cells are concerned, well, they do not know each other. That is, as I have already said, that adult somatic cell hepatocyte do not recognize your adult myocyte or cardiomyocyte. Therefore, if all the physicians are genetically similar, their attitude towards proportion, all the doctors are doctors, but a physician is different from a gynecologist. How is it so? Because it is genetically they are similar, but functionally they are not similar. A physician is not a gynecologist because he was forgotten. And how does it happen? One is known to each other. Even all physicians are genetically similar. Their attitude towards proportion depends on the environment. Now comes the environment. That was dependent on the gene. Gene has made them. It was the factor of gene. I said in the development of the somatic cell, there are two factors involved. One is the gene, which makes a doctor. But there is an environment. If you are in contact with a liver cell all the time, throughout the time, your attitude will be moved towards liver. Because if you are in contact with sadhu for a longer period, you are bound to become, if not totally sadhu, partially sadhu. And if you are a dacoit, where you are not totally dacoit, but you will live. So that is environment. The cell living in close environment with other cells which are behaving in one way compared to the other group of cells which are behaving in some other way will be different. That is what is known as media in which you are growing, or some stimulus which we do not know. And that is the reason why stem cell culture research has failed. We do not know which stimulus or which signal is making a cell which is similar to the cell like heart, which is also making a cell like heart into a liver. So there must be some difference in the stimulus. and that is the reason why a heart cell is not becoming or not able to become a cardiomyocyte. Therefore, even all physicians are genetically similar. Their attitude towards profession depends on the environment, more academic, more clinical, in which the individual physician has been reared out. This determines whether he or she will be a clinician. That stimulus, that signal, and that determines whether he or she will be a clinician, basic medical scientist, or a clinician scientist. So there are three groups. After a student has been admitted to the medical curriculum, scientist, basic scientist, or a clinical scientist, or a clinician. So that will depend in the environment in which he has been grown up. He or she has got the medical degree into a clinical doctor, or a clinical scientist doctor, or a basic scientist doctor. So this environment we are trying to find out in stem cell research. That is what stimulus will just drive this cell towards the heart, what stimulus will try to drive this into a pancreas, or what stimulus or what trigger will drive this. Just like a media, Ratna Chakravarty is keeping the cell in different media with, so that will determine whether it will grow into an adult cell, or it will be damaged, or it will be semi-damaged. Now in ART, this is exactly what is happening in ART. That is, the clinician treats infertile couple. There was no understanding of the oogenesis or fertilization. When ART did not come in, and ART did not, artificial reproduction did not come in. People know there is reproduction. Is what that, and because she is a woman, she is a man, they live together, and therefore they have produced the baby. And how this, where from this baby came, whether what was spermatozoa, what was oocyte, that was not known. So there is no understanding of the oogenesis, not to speak about where from it came about. Now this oogenesis, unit of birth in the female or unit of birth in the male, that is the spermatogenesis, was not known. Till you have to know, you have to try to know. That is, the clinician decided to turn into a clinician scientist, because he is delivering the baby, but he doesn't know how the baby grows. So wanted to think. Bob Edwards wanted to think when he was possibly 40, when he was 45. Then how this baby comes in, nobody knows. But he started thinking that where from it comes, what is spermatozoa, what is oocyte, and after that, where from oocyte comes, how does it oocyte develop, how does the spermatozoa develop. So that is why this was the term used till the clinician decided to turn into clinician scientist. So unless you try to think it, how do you know? You cannot do it. And therefore it was possibly people like Bob Edwards trying to think that where from the baby comes and what is spermatozoa, the terms usually used to come in, and what is your oocyte or oogenesis. So clinician scientist is the person who has the potentiality to think like that. Why it is happening? Unless, so science will never develop, science will never stand. So that is how the thought came in the human body's mind. This clinician scientist was able to understand the process behind, because the disease you know, but by which the disease is occurring you don't know. So they tried to think about that. Clinician scientist was able to understand the process behind, and hence the process of IVF and ART. And that was the origin of IVF and ART. Also long before that, I think it was in 1700, people started thinking, but that was materialized in 1952 with birth means scientific birth of Bob Edwards. And he started to think that what is oogenesis, how does ovum develop. So this is in vivo. He tried to think in vitro. That is, in the laboratory, from animal experiment he used to know. So that is how in vivo he knew, but how in vitro it can be done. He could understand the procedure. So this is known as in vivo oogenesis and fertilization. This is on the left hand side, in vivo oogenesis. That is the primordial follicle secondary follicle, all those things, he came to know. And then in vitro, he tried to think how this is the fertilization. Now he knew oogenesis, he knew spermatogenesis, but how does fertilization occur? Make 2 into 1. 2 into 1 means oocyte and spermatocyte united one cell, and that is zygote. So how the process came in, so he became a clinician scientist. So this process was not known before that. So it was in 1952, I think it became successful. Bob Edwards came and described this process of oogenesis and fertilization. So these are the processes, the picture depicts in vitro oogenesis and fertilization in petri dish, almost similar to in vivo. In petri, how to do it? How could we mimic the original process inside the human body? And that is a clinician scientist. So that speciality or that originality he had, and therefore he became a clinician scientist. Had the clinician not decided to turn into clinician scientist, IVF would not have developed. And so this instinct came from that in vitro fertilization in the beginning when he was in the mother's uterus, mother's womb. So by a process which is known as continuous research in this field helps us, and from that time on, continuous research in this field helps us in providing the success rates in stimulating the in vivo process as close as possible. Now, this is when I thought back, I think this is the way in which I have turned up to the position where I am today. And I am very grateful to NRS Medical College, although I was not born there, I was brought up there. It is just a similarity in between. When thought back, that this is the similarity of my career. So long, 60 years I have passed through this process, and then I have been able to establish myself in the position where I am standing today. So I was born in Calcutta Medical College, but I was brought up in NRS Medical College. Whatever I have achieved, big or small, has been possible entirely because my long association, maybe around 20 years with this prestigious institute. So that is how I think when I started thinking about myself at the end of my days, that how could I come there? So I said my career is also just like that. At the last end, ultimate days of my career or whatever lifetime longevity, well, I have gathered this one through the same process as Steptoe and Edwards, where everybody, I mean, who has turned into this different, everybody is a doctor, but everybody is a clinical doctor. So my first phase was 1952, I graduated, and from there MO, MRCOG. Ambition was only to go, if you tell somebody, some junior, ambition was to become a good doctor and a good clinician, not beyond that. I mean, whether I will be a researcher or I will be a clinician was the ultimate fate today into where I am today. Now in phase 2, 62 to 68, I earned reputation as a skilled surgeon and a good teacher. But some of my senior professors at that time criticized me and remarked only good batting, I was good at operation, but I was not publicizing anything. I was making any publication. I was not in the academic field. Some of my senior professors, Professor K.N. Mithro, remarked only good batting is not a criteria of good cricketer. You must learn fielding as well. And what do you mean by fielding? That is collecting the data, collecting case reports and publishing. Unless you do that, you will not be a good cricketer or good doctor. And that is how you should collect data of new projects and publish papers. And there my habit of collecting data came in. I had a special craze for dealing with uncommon patients with ill-defined treatment modality and obviously with unfavorable outcome. And that is the reason why initial phase my crude surgical skill had turned into a finer surgical modalities. I worked extensively on surgical repair of vesicovaginal fistula. At that time, they were usually thrown away by surgeons. Pelvic exentration thrown away by surgeon for advanced carcinoma of cervix, and finally landed on final surgeries like tuboplasty and surgical repair of congenital absence of vagina and uterus, resulting in delivery of viable babies. Published papers attracted visitors from abroad who came to see my vaginoplasty and hysteroplasty at NRS Medical College. And that is the way in which I stepped up in profession to some extent. Not that I have done everything, but to some extent whatever I have done only by this instinct. And this instinct means association. There are two things. One shows my instinct to become a special doctor, and secondly, I had the association to do that, and I got the association. Published papers attracted visitors from abroad who came to see my vaginoplasty and hysteroplasty at NRS Medical College. And that is the way in which I passed my stages in medical curriculum, and these are the types of operations that I used to do. Phase 4, 1975 to 1982, started work in collaboration with Dr. Subhash Mukherjee, a brilliant scientist, Department of Physiology, NRS Medical College, environment. This brought me a new change of environment, and with change of environment from dacoit, I think I was becoming gradually a sadhu. So NRS Medical environment to become a clinician scientist. Worked extensively on infertility, endocrinological, genetic and developmental disorders. Published papers on testicular feminizing syndrome, 46XY sex reverse female, 47XXY female testicular Klinefelter syndrome. All these papers I published. Only I got the incentive from my neighbors and from the association that I got at that time. This has happened. I never thought that I would do it, but this has happened in my life, and that is the reason I have published so many papers. I am not proud of that. I am not boasting for that, but this has happened in my life, and that is the reason why I have done a few things in my life. So these are the ART work, and these are the clinical papers Subhash Mukherjee brought it, and this will be, I think demonstrated in slide form. And ART work started in 1983 and still continuing. Fortunately and unfortunately, I am still surviving and continuing to some extent, contributing to some extent. Incentive from Dr. Subhash Mukherjee, pioneer of IVF work in India, experimental work in many areas of human reproduction, work being conducted in collaboration with IIT Kharagpur, CCMB Hyderabad, IICB Jadavpur and Kolkata. And the last few words I will pass on to my juniors. And a physician's primary duty is to work with a stethoscope. But this work pattern is to be improvised by the information gathered with the help of a microscope. This is possible only through bridging the gap between clinical medicine and basic science. In conclusion, what we want to say is what we are today is the result of our own past actions. What we wish to be in future depends on our present actions. Decide how have to act now. We are responsible for what we are. Whatever we wish ourselves to be, we have the power to make ourselves. Thank you very much for your patient hearing.